processes are those types of transport that allows cells to move things into and out of themselves across their membrane. But these types of transport processes require ATP. That's what active means. It allows us to do things that the universe doesn't normally do. It allows us to go backwards against a concentration gradient. We can use it to maintain a differential in concentrations on different sides of the membrane. And by doing that, we can build up a source of readily available energy. Primary active transport is using energy directly from the breakdown of ATP. This is phosphorylation. So we're going to take that third phosphorus on the adenosine triphosphate, and we're going to break it off, giving us adenosine diphosphate and that third phosphorus that we've just broken off. That third phosphorus is then added to the transport protein, and it's like plugging in a battery. And that protein can now change shape and force movement to happen across the membrane. So an example of this is an ion pump. There's little cellular proteins that pump ions across the membrane. Pump implies that we're going to use energy, it's an active process, and we're going from a low concentration to a high concentration. So this is backwards from diffusion. So for example, in muscles, we need to store calcium ions inside a little membrane-bound vesicle, little membrane bubble inside of the cell, and we need to collect all of the calcium in the cell and get it into this membrane. Now, the universe doesn't like to collect things together, right? It likes to spread things out, which is what diffusion is. So by using active transport and calcium ion pumps, we can grab spare bits of calcium, spare ions, and push them into the vesicle, even though the concentration inside is higher than it is outside. One really cool example is called the sodium-potassium pump. Now this is what's called an exchange pump. That means as we pass one ion in one direction, we're going to pass the other ion in the other direction. We're going to be moving both of them against their concentration gradients. The nifty thing is we're going to kind of cheat just a little bit here, and we're going to move 3 sodium and 2 potassium ions, all for the price of just 1 ATP molecule. So here's our sodium-potassium pump. We're going to start off here with number 1. You can see the blue here is going to be inside of the cell, the cytosol, and this area to the left is the interstitial fluid, or outside of the cell. Here's our phospholipid bilayer. We've got our polar heads and our nonpolar tails. And we've taken 3 sodium ions, and they're going to fit into special spots inside the transport protein. Those sodium ions lock in because those spots have the exact correct shape for sodium ions to plug in. Adenosine triphosphate will come along, phosphorylate the protein. So we're going to pull that third phosphorus off, and now we're left with this adenosine diphosphate molecule that can go away. And that third phosphate, which has been stuck onto the protein, and this causes the protein to literally change shape. And as it does so, those little binding sites for the sodium ions are going to change shape, and the sodium no longer fits. So the sodium falls out, and the only place it can go is out of the cell. Now at the same time, those sodium ion docking spots change shape to no longer allow sodium to fit. We have some other spots that opened up for potassium ions. So down here, potassium ions can now fit into the protein. The sodium has moved outside of the protein. The phosphorus, that inorganic phosphate that we had on there, will detach, and that is the trigger to cause the protein to move back to its original shape, meaning that the potassium ions no longer fit, and they have nowhere to go except inside of the cell. So for the price of 1 ATP, we're going to continually cycle 3 sodium ions to the outside of the cell and cycle 2 potassium ions to the inside of the cell. This little protein is so fundamentally critical that you spend a third of your caloric intake every day to making enough ATP to run the sodium-potassium pump in your body. That's a third of your calories just to run this little pump that you may never have heard of before. That's amazing. Now, secondary active transport is a little bit different. We're going to use primary active transport, the one I just talked about, to set up a concentration gradient to give us the power to go ahead and do another type of active transport. So it sounds a little confusing here, but we're going to, it's like driving a car to the top of a hill and parking it. It takes energy to get to the car to the top of the hill. You've already spent the energy to do that, and then as you take the car out of gear and give it a push, it's going to go ahead and roll downhill because of gravity. So with secondary active transport, we're going to use primary active transport, an ATP, to set up a concentration gradient, which will let us get to the top of the hill. And then as we allow the ions to diffuse back to where they would normally go, that's the car rolling down the hill. So it lets us get further along in our biochemical processes without spending as much energy, because we use some of it to kind of set up that primary, and then the secondary kind of happens automatically. So as we do secondary active transport, we could do it two ways. One is called symport, the other is antiport. Symport is where the two ions are both moving in the same direction, either both into or out of the cell. And antiport is where the two ions are moving to opposite sides of the membrane. So here's a concrete example for you. We've got our sodium-potassium pump. Now, the sodium-potassium pump never shuts down. In fact, it keeps going for several hours after you're dead. As long as there's ATP, the sodium-potassium pump will just continue to work. So we've taken that sodium-potassium pump, we've built up this large concentration of sodium ions outside of the cell and very few sodium ions inside of the cell. Diffusion would love to spread those sodium ions out, if only they were allowed to come in. So that's where we use a symporter. This is a special protein. It doesn't use ATP. We used ATP with the sodium-potassium pump to push all of the sodium out of the cell. But what the symporter does is it will allow the sodium to diffuse back in, but only as long as it takes something in with it. And that particular something is going to be specific to every kind of symporter. In this case, this is a glucose symporter. So we have to use the sodium-potassium pump to pump sodium ions out of the cell, and then sodium can diffuse back in, but only if it brings glucose with it. So we set up our primary active transport, building up the concentration gradient, like driving the car up the hill, and then as the car rolls down the hill, or in other words, the sodium diffuses back into the cell, it can only do so if glucose comes too. And so this allows us to bring glucose from a lower concentration to a higher concentration, which is again active transport. It's just we're not doing it directly. We're not driving down the hill. We're just coasting down the hill. And then to the right here, we have our antiport. This is showing us how potassium will move one way, but only if hydrogen moves the other. Now, if we back off just a tiny bit, we can get into vesicular transport. So this is using a large area of the plasma protein. This is not using just a single protein transporter. So this is called bulk transport. We've got two basic varieties here. We've got exocytosis and endocytosis. Exocytosis is the cell moving something outside of itself. So large molecules, macromolecules that are too big to go across the membrane through diffusion or too big to go across the transport protein, we're going to use this vesicular transport and the process of exocytosis to move stuff out of the cell. On the left, we have the cytosol, the inside plasma of the cell. Here's our plasma membrane with our phospholipid bilayer. And then on the right, we've got our interstitial fluid. So we've bound up some product here, these little tiny boogers, inside of a membrane bubble called a vesicle. Again, it's just made out of phospholipid bilayer. As that membrane bubble gets towed to the surface of the cell, it's going to then merge with the surface of the cell. It's going to merge with the phospholipid bilayer of the cell membrane, and then it's going to just kind of even out, and as it smooths out, that vesicle membrane becomes incorporated into the regular bit of the plasma membrane, and what was inside of the vesicle is now outside of the cell. Endocytosis is the exact opposite. Endocytosis is bulk transport, where we're going to use a large portion of the plasma membrane to grab something and bring it in. We use this for all kinds of stuff. Materials for digestion, white blood cells will do this in what we call phagocytosis, which is a specialized kind of endocytosis. This allows the white blood cells to eat things like bacteria. First, we're going to do what's called invagination. We're just going to take an area of the cell membrane. It's going to sink down into the cell underneath the thing we're going to bring in. The pocket gets deeper and deeper, and it will slowly pinch off, and then you have a bubble inside of the cell. So going back a couple slides here, it's this exact process, but in reverse. We're going to have that membrane sink down a little bit, and as it continues to move into the cell and get deeper, the outside opening is going to go ahead and pinch off, and now we have a vesicle of substance that we've grabbed from outside of the cell, but now it's packaged up and inside of the cell. So I know most of the time students have this perception of the cell membrane being very static, very stable, and not really moving a lot, because it doesn't move in the pictures and it doesn't move in the plastic models you see in lab, but it's extremely dynamic. It's massively busy. There's stuff moving back and forth across that membrane constantly. And not only that, not only do we have diffusion and active ionic transport, but we also have this bulk transport where entire sections of the plasma membrane are caving in and sinking down into the interior of the cell, and in other places, big portions of membrane are coming up out of the depths of the cell and bubbling up and then incorporating themselves into the outside cell membrane. So cell membranes are massively, massively dynamic. As I mentioned, phagocytosis is a specialized type of endocytosis. Phagos is to eat, and cyto would be cell, so it's cell eating. And not a lot of cells do this. It's pretty much our white blood cells, and they do this to eat bacteria. So here's an example. Here's our cytosol and our plasma membrane. The white blood cell will actually grow these little pseudopodia, these false feet, around a particle, whether it's a bacteria or bits of debris, dead cells, and those two pseudopodia are going to kind of meet together, and then we have a pinched off vesicle that's now inside of the white blood cell. The white blood cell can then fairly easily destroy the bacteria. Pinocytosis is cell drinking. This is kind of rapid scale endocytosis. The membrane is just taking little gulps of the surrounding area. And then we have something called receptor-mediated endocytosis, where there's going to be a specific docking protein on the surface of the cell membrane. And once the correct molecule docks into that, that's going to cause our endocytosis to occur. There's several steps here. We'll just kind of skip over to it. We've got these docking proteins called receptors, and then there's specialized proteins called clathrin that kind of surround part of the membrane. And as the docking proteins receive their molecule and are triggered, that's going to cause that endocytosis to happen. So to kind of recap here, we have our passive processes. We've got our simple diffusion where things go back and forth. And remember, in order for things to diffuse across the cell membrane, they have to be lipid soluble or nonpolar or hydrophobic. It's the same thing. All those words mean the exact same thing. And we're just going to simply go from a high concentration to a low concentration until equilibrium is established. Facilitated diffusion is for things with a charge, things that are polar or hydrophilic. They cannot diffuse through the cell membrane, so they're going to have to use a doorway. So this is our facilitated diffusion. These proteins will facilitate or help with the movement of these charged particles. And then lastly, osmosis is specifically the diffusion of water. Our active processes, we had our primary active transport, and then we have secondary active transport, and then our types of vesicular transport, exocytosis, and then a couple varieties of endocytosis. There was pinocytosis, phagocytosis, and receptor-mediated endocytosis.